Good morning to all. Hello to everyone joining today's webinar. I'm Dr. Pradeep Golani, and as the co-convener of this event, I'm delighted to welcome a diverse and engaged audience. Your participation greatly enriches our discussions and underscores our shared commitment to advancing in our respective fields. I look forward to productive conversations and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in pharmaceutical sector. I am confident that today's session will uphold our tradition of excellence, providing valuable insights from a respected speaker. Let us seize this chance to gain new knowledge, participate actively, and progress our field. Welcome, everyone. I look forward to a productive experience for all participants. Thank you for joining us today. We are gathered today to commemorate the 88th SPSR International Webinar on microRNA regulation of diabetes associated atherosclerosis, a distinguished event hosted by the Society of Pharmaceutical Sciences and Research. Under the esteemed guidance of Dr. Shashi Alok, National President of SPSR, the Society continues to play a pivotal role in fostering innovation and advancement in the pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy-related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It is our privilege to introduce Dr. Shruti Rawal, faculty, Brigham and Women's Hospital, Harvard Medical School, Boston, USA, as a distinguished speaker. Dr. Shruti Rawal expertise greatly contributes to our event, providing substantial knowledge and insights. Her participation is certain to enrich our discussion and expand exploration of microRNA regulation of diabetes associated atherosclerosis. We are grateful for our engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monika Sabharwal as the convener for today's webinar, who also serves as the founder and National Secretary of SPSR and CEO SPSR Periodicals Private Limited. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session. I am pleased to invite Mrs. Monika Sabharwal to warmly welcome our distinguished guest speaker. Over to you, ma'am.
Thank you so much, Dr. Pradeep Golani. Hello, everyone. I, Monika Sabharwal, on the behalf of Society of Pharmaceutical Sciences and Research, SPSR, I extend my heartfelt welcome to all the participants, academicians, industry professionals, students, and researchers from across the globe to the 88th SPSR International Webinar on highly insightful topic microRNA regulation of diabetes associated atherosclerosis. We are truly honored to host Dr. Shruti Rawal ma'am as esteemed faculty member at Brigham and Women's Hospital, Harvard Medical School, Boston, United States of America, as our distinguished speaker for today. Her cut-edge research and the global academic contributions in the field of molecular biology, and the cardiovascular disease are immensely inspiring, and we look forward to learn from her expertise. We are proud to share that this webinar has garnered over 1,600 registrations from more than 20 countries, which reflects the growing interest and the trust in the SPSR's mission to foster knowledge sharing in the pharmaceutical and biomedical sciences. Today, as we celebrate the 88th webinar in this journey, I wish to acknowledge the continuous global support that have been driving force behind our growth. Our audience positive feedback, active participation, and valuable suggestions motivate us to raise the bar every time. More than that, 98% of our attendees have shared that SPSR webinars have positively impact their professional journey. And with this, what keeps our team going even more energy and with a purpose. At SPSR, we strive to bridge the academia and the industry by regularly inviting speakers from both the domains, encouraging the collaboration and deeper understanding. Our webinars are conducted completely free of cost to all of you, allowing access to the high quality knowledge share for students, researchers, and the professionals alike. This year, SPSR has expanded its reach through national and international collaborations, faculty development programs, and the growing partnerships with the reputed pharmaceutical institutions. We are proud to inform that our SPSR YouTube channel has crossed 21,000 subscribers from 50 countries, and our social media following is rapidly increasing. It is encouraging to see how our efforts are trending globally. We are also delighted to welcome the increasing numbers of professionals who are joining the SPSR membership program, helping us to build a stronger network of knowledge seekers. If you are interested in joining our SPSR family, we invite you to explore the membership details on SPSR's official website. I want to express my deepest gratitude to all the participants for joining us, especially those who make a point to attend regularly every Sunday. Your consistency and the passion fuel our commitment to quality content and impactful learning. Before we commence, I would like to warmly welcome our National President, Dr. Shashi Alok, whose visionary leadership is very crucial in the impressive transformation of the SPSR. I would like to extend my heartfelt thanks for the significant contributions made by Dr. Pradeep Golani, the co-convener of this webinar, and the organizing committee members, Dr. Saurabh Kose, Dr. Sanjay Nagde, Mr. Ankur Agarwal, Mr. Junaid Tantre, and Mr. Rishabh Manor. Their tireless efforts have played a crucial role in transforming vision of this event into a reality. Join the SPSR vibrant community of over 50,000 pharma professionals actively engaging across LinkedIn, YouTube, Facebook, and WhatsApp. Let's connect, learn, and grow with the top tier webinars and the exclusive content every time. Do not miss out this unparalleled opportunity to elevate your pharmacy practice. Stay updated by subscribing the SPSI YouTube channel and enabling the notification bell icon for the instant alerts. Once again, I extend my heartfelt gratitude to our distinguished speaker, Dr. Shruti Rawal ma'am, for joining us today and the sharing her valuable insight with us. I also sincerely thank each one of you for being part of ever-growing SPSR family. Together, let us continue to advance the knowledge, empower professionals, and elevate the global pharmaceutical community. With a great enthusiasm, let us now commence today's scientific journey. Thank you and welcome once again to the 88th SPSR International webinar. Thank you, and over to you, Dr. Pradeep Galani.
I appreciate Mrs. Monika Sabharwal granting me the opportunity to present this webinar. And now we will start with the short introduction to guest profile. Dr. Shruti Rawal is a distinguished cardiovascular scientist with a deep-rooted expertise in pharmacology, molecular biology, and immunometabolism, focusing on uncovering the immune metabolic mechanism that contribute to heart disease and diabetes. She began her academic journey in India, completing her B.pharmacy and M.pharmacy in pharmacology from Guru Nanak Dev University, Amritsar in 2008. Between 2009 and 2012, she served as the faculty member at the School of Pharmacy, Lovely Professional University, where she taught undergraduate and postgraduate students. Her passion for research led her to pursue a PhD at the University of Otago, New Zealand, where she explored the pathological roles of cardiovascular microRNAs in diabetic heart disease. In 2017, she moved to the United States to undertake postdoctoral training, further expanding her expertise in RNA biology, immunology, and metabolomics. Now a faculty member at Brigham and Women's Hospital, Harvard Medical School, Dr. Rawal was promoted to the role of research scientist in 2022. And in 2024, she began leading her own research group. She is currently supported by the American Heart Association Career Development Award, which enables her to investigate the regularity roles of microRNAs in inflammation and vascular dysfunction associated with type 1 and type 2 diabetes. Her lab utilizes a multidisciplinary approach that integrates RNA transcriptomics, flow cytometry, murine models of atherosclerosis, and metabolic profiling to discover novel therapeutic targets aimed at preventing cardiovascular complications in diabetic populations. Dr. Rawal has published her findings in several prestigious journals, including Nature Immunology, Cardiovascular Research, and Clinical Science. She is also actively involved in mentoring young scientists and promoting interdisciplinary collaborations with a strong commitment to translating basic scientific discoveries into clinical solutions. With her pioneering work and leadership, Dr. Shruti Rawal is at the forefront to cardiovascular research aimed at improving outcomes for patients with diabetes. It is with great satisfaction and pride that we declare that we have exceeded 1,600 registrants globally, showcasing an impressive diversity of countries. Significant attendance is noted from the nations including India, Angola, Bangladesh, Ethiopia, Indonesia, Iraq, Kenya, Kuwait, Malaysia, Mexico, Nepal, Nigeria, Congo, Saudi Arabia, Sri Lanka, Uganda, USA, and Uzbekistan. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Shruti Rawal, with us. Welcome, ma'am. Your insights would be highly valuable, and I would appreciate if you could present them in a speech. Over to you, ma'am.
Thank you so much for such a warm welcome. And I'm so excited to present my work at this large platform. My work mainly involved studying the role of microRNAs, understanding the mechanistic as well as the therapeutic role of microRNAs in context to diabetes associated atherosclerosis. So let me first introduce my journey, where I started and where I am um, today, some of which you have already listened. So I did my pharmacy, B.pharmacy from Kurukshetra University in India, and then my master's in Amritsar, GNU, and my master's was mainly in diabetes research, which is where I started growing interest in this field. And then I went for teaching at LPU for 3 years and was also supervising some master students with the focus in diabetes research. I continued this interest and then went ahead to do my PhD in University of Otago, where I secured a full scholarship for throughout my PhD, and my PhD was mainly to study the role of microRNAs and diabetic heart disease. And then after completing my PhD, I moved to United States to join Dr. Fisher Lab at New York University in New York, where I first started to learn about atherosclerosis, which was a great platform for me to introduce into this field. And then next, I joined Harvard Medical School at in the Charas Lab, which was an industrial research fellow position in 2019, where I developed expertise in the field of immunology, which really helped me to then move to my next level, where I joined the Feinberg Lab, which was mainly working in microRNAs and atherosclerosis. So I was able to integrate my prior knowledge in the field, as well as learning more about the non-coding RNA biology. This is where I then moved to the next stage of my career, where I was promoted as a research scientist in 2022. And then I was also awarded with the American Heart Association Career Development Award in 2024, which helped me to be promoted as faculty in Harvard Medical School, where I am currently pursuing my research with. So entire journey of my career has helped me to have expertise in diabetes, in microRNAs. It's also evolved around cardiovascular diseases, more specifically in atherosclerosis and immunology, which is today's topic, microRNA. Just a brief introduction about microRNAs. So these are small non-coding RNAs that are mainly transcribed in the nucleus through the DNA sequences, where they are first formed as primary microRNAs and then cleaved from DROSHA RNA binding enzymes to precursor microRNAs, which are then around 70 nucleotide long. And these precursor microRNAs are then exported to cytoplasm, where they are then formed as duplex and ultimately make as a mature microRNA. And what the function of this mature microRNA is to bind to the mRNA or genes and therefore regulate protein production. And there are three main mechanisms by which these microRNAs regulate the gene expression. Most prominent is they bind to the 3' UTR region of the genes and causes the mRNA degradation. They can also cause translational repression, and they can also sometimes cause the cleavage of their mRNA targets. And by doing these mechanisms, these microRNAs can fine-tune the expression of certain targets or proteins and therefore regulate several biological processes. To our interest is the cardiovascular diseases. There are around dozens of microRNAs that have been studied in various fields of cardiovascular diseases. to my interest, I am interested in atherosclerosis, where there are three microRNAs that are popularly being studied: microRNA 155, microRNA 21, and microRNA 33. And I'm also going to introduce another microRNA that I am currently working on in this field. Just a brief introduction about what is atherosclerosis. So this disease is mainly characterized by accumulation of lipid-laden macrophages. Those are immune cells in the subendothelial layer of the aorta. And this accumulation is more advanced in people with diabetes, which leads to a higher burden of this disease in diabetic subjects as compared to the non-diabetic subjects. However, what mechanisms drive these processes in diabetics as compared to non-diabetics is still not clear. So when I joined Feinberg Lab, I was very interested in understanding how non-coding RNAs, particularly microRNAs, regulate these processes. So the first question I asked was, which microRNAs are dysregulated during the progression of diabetes-associated atherosclerosis? And to address that, we performed an extensive animal study where LDLR knockout mice were fed on high-fat sucrose-containing diet. And just to introduce this diet will mimic a human diabetic condition where it will have high glucose as well as insulin resistance in these mice over a period of 12 weeks of diet feeding. After 12 weeks of this diet, we isolated the aorta from these mice, then peeled their intima, and then subjected those to the RNA sequencing, particularly the microRNA sequencing. And from this special microRNA sequencing, we found that there were 137 differentially expressed microRNAs. And among these differentially expressed microRNAs, one microRNA that was top among the down-regulated microRNAs was microRNA 369-3P. So we validated these observations through PCR between the control as well as the advanced atherosclerosis. So we took both intima and the media sections of the aorta and looked at the expression of this microRNA. What you can really appreciate is that with the advancement of atherosclerosis, there is a significant decrease in the expression of this microRNA, validating what we found was in the RNA sequencing. And another observation from this experiment was that the expression of this microRNA was more in intima as compared to the media. And as a reminder, intima consists of cells like endothelial cells and also leukocytes, particularly monocytes, macrophages that come and then deposit in the intimal layer. So this was very promising. And then we also wanted to understand the translational aspect of this microRNA, because this microRNA has not been studied in any of the inflammatory diseases before. So what we did, we took help of our collaborators at Boston University Hospital, where they were collecting human PBMCs from people that have diabetes, those that didn't have diabetes, but coronary artery disease, and those that have both diabetes and coronary artery disease. And what we found was that the expression of microRNA 369-3P was significantly decreased in patients with diabetes. But what was more profounding was that people with diabetes and coronary artery disease had further downregulation of this microRNA as compared to the counter non-diabetic and coronary artery disease patients. So having translational aspect established, we next wanted to understand what particular cell types express this microRNA. So we did an extensive cell type profiling. Here are some of the cell types that I profiled, and we found that this microRNA was highly expressed in the immune cells, particularly the macrophages and the monocytes. After that, we also wanted to understand its expression under different stimuli, such as the pro-inflammatory stimuli like LPS, ox-LDL, palmitate, IL-1 beta. All of these stimuli are known to enhance the pro-inflammatory status of macrophages. We also looked at its expression under diabetes, that is high glucose, HG, as shown in the second bar. And we found that with diabetes as well as with the inflammatory stimulus, the expression of this microRNA was significantly decreased. However, when we polarize these macrophages towards the anti-inflammatory phase through IL-4 stimulus, there was a restoration of this expression, suggesting that this microRNA was having a role in the inflammatory and diabetic condition in macrophages. So with these preliminary results, we hypothesized that microRNA 3693P regulates macrophage inflammation. And secondly, therapeutic administration of this microRNA can halt the progression of atherosclerosis in diabetic mice. So it is well known that oxidized lipoproteins are known to cause metabolic reprogramming in macrophages by increasing glycolysis while decreasing mitochondrial respiration and oxidative phosphorylation. And by doing so, the mitochondria then generate a lot of reactive oxygen species, which then activate inflammasome components and therefore propagate inflammation in these cells. So taking oxLDL as our inflammatory stimulus, we then asked whether if we overexpress 3693P in the macrophages, can it mitigate the oxLDL-induced mitochondrial dysfunction, and also can it restore the cellular homeostasis? To answer this, we performed seahorse assay on oxLDL-loaded bone marrow-derived macrophages. And what we read from that assay was the mitochondrial respiration as well as the glycolytic rate of the macrophages. So first, when we looked at the mitochondrial respiration, you can really appreciate that the bar in red color, that is those control cells with only oxLDL, that have significant decreased mitochondrial respiration, while the cells that were overexpressed with 369 mimic but subjected to oxLDL stimulus still was able to have a higher mitochondrial respiration. We also then quantified each component of mitochondrial respiration. In the right panel, you can see that cells that have overexpression of 3693P were able to have increased basal respiration, maximum respiration, increased ATP production while decrease in the proton leak, suggesting that the overexpression of this microRNA was able to restore the mitochondrial respiration that was otherwise being attenuated in oxLDL-loaded macrophages. The second aspect of this study was that when the cells were only loaded with oxLDL, as shown in the red bar, you can see there is increase in glycolysis. While when the cells were overexpressed with microRNAs, this glycolysis was then shifted down, suggesting that this microRNA was able to mitigate the glycolytic activity in these cells by by regulating the immunometabolic role that was further the question of investigation for our study. Because the mitochondrial respiration was restored, we were very interested to see if the oxidative phosphorylation was also restored. And to our interest, we found that most of the components associated with oxidative phosphorylation were all restored when the cells were loaded with 369-3P overexpression. Most prominent effect was on the complex 2, if you can see in the bar graph, which is also associated with the Krebs cycle, or the most of the TCA metabolites are being produced under this subunit. So because oxLDL is known to mediate mitochondrial dysfunction, it is also known to disrupt Krebs cycle and therefore accumulate and release pro-inflammatory metabolites. We were very interested to understand what kind of metabolites were being accumulated and if they were also being restored by 369-3P overexpression. So we performed mass spec in PMDMs. What we did, we isolated bone marrow-derived macrophages from wild-type mice and then overexpressed microRNA in these differentiated macrophages. And after 48 hours of overexpression, we then introduce ox-LDL for 24 hours to introduce a inflammatory environment in these cells. Thereafter, we collected the cell lysates as well as the supernatant to perform the mass spec. And these two type of samples were then used to study the intracellular as well as the extracellular metabolites under different conditions. So what we found was that among the differentially regulated metabolites between the oxaldia-loaded macrophages as well as those that were treated with microRNA, there were 13 commonly common metabolites. But what really caught our attention was one metabolite in particular, that was succinate. That was the top metabolite being regulated by microRNA 3693P. What was interesting about this metabolite was that it was highest increased in the cells loaded with ox-LDL, but when this microRNA was overexpressed in these cells, there was significant decrease in the amount of this metabolite being released. Then we also looked at these metabolites for the pathways they were associated. To our interest was succinate, which was associated with several metabolic pathways as listed here, including the TCA cycle. So then we wanted to understand the role of succinate in propagating inflammation in macrophages. So what we did, we took the primary monocytes from the wild type mice, and then we primed them with succinate. Even the prior studies have shown that just succinate itself is not enough to stimulate the cells towards a pro-inflammatory environment. So therefore, we primed the cells with succinate and then followed the treatment with ox-LDL. What we found was that after priming the cells with succinate, there was huge increase in the pro-inflammatory subset of monocytes, which is Ly6C high, while there was a significant decrease in the Ly6C low subset of monocytes in these cells, suggesting that there was a shift of the status of these cells towards a pro-inflammatory environment as compared to the anti-inflammatory environment. Another interesting observation of this experiment was that while oxaldia loading increases the IL-1 beta secretion in the supernatant of the cell of these cells, there was no increase with the succinate itself. But when the cells were primed with succinate and then loaded with oxaldiol, there was a huge increase in IL-1 beta secretion. So together, this suggests that succinate priming increases the oxaldiol-induced pro-inflammatory phenotype in the mouse primary monocytes, something that we can depict might also be happening at the in vivo level. Next, to understand further depth of the succinate, we also looked at its intracellular levels. What we found was the cells that were loaded with ox-LDL that had huge increase in the accumulation of succinate intracellularly. While when we overexpress our microRNA, that increase was then mitigated, suggesting that 369 overexpression was able to decrease the accumulation of succinate in the intracellular level. Previous work from Luke O'Neill has also shown that the intracellular succinate accumulation activates HIF1 alpha signaling. So we were quite interested to understand under ox-LDL conditions, does these pathways are also regulated? So what we found was that with ox-LDL, there is huge increase with the HIF1 alpha expression, while when 369-3P was overexpressed, this HIF1 expression was significantly decreased. And when we next wanted to understand if this decrease in 369-3P expression was through succinate decrease, we use DMM, which is a succinate inhibitor. And when we introduce a succinate inhibitor, that increase in ox-LDL was not reported, was not observed as compared to the graph shown in the middle, suggesting that the increase in HIF1 was relying on succinate accumulation, and when we inhibit that, there was that accumulation was decreased, and therefore the HIF1 activation was no longer there in these cells. Because succinate emerged as one of the main metabolite in our study, we were very interested to understand further its pathological role in diabetes. So there was one study that showed that under high glucose, there is a release in succinate from the keratinocytes. And what was important here to note that this metabolite has a potent receptor known as GPR91 or succinate receptor 1. Where it binds to its receptor and then activates downstream inflammation pathways such as NF-kappa B pathway. And there were other studies that also showed that this pathway further downstream also activate inflammasome signaling. So we were quite interested to understand what signaling is being regulated in this environment and if microRNA 3693P can regulate these signaling. Parallelly, our bioinformatic team was also looking into the targets for this microRNA. And very interestingly, we found that succinate receptor 1 or GPR91 is also one of the targets for microRNA, which was shown. If you see here, we first took those targets that were putative in the mice, and then those targets that were putative in the human, and then from there we took 303 targets that were common between the mouse and human. So we were interested in the conserved targets for this microRNA. We took this approach further and then looked at what these targets were associated at. There were several pathways that were associated with these targets, but we were mainly interested in those targets that are involved in regulating inflammation. So from those 303 targets, we narrowed down our search to 7 targets shown here. And among those 7 targets, one target that caught our attention was succinate receptor 1, which is also known as GPR91. So with this discovery, then we further validated that it is a direct target through luciferase reporter assay, where the cells were overexpressed with microRNA 3693P and also with the GPR91 plasmid. When you overexpress this microRNA, the luciferase reporter activity of this wild-type construct was significantly decreased. However, when you mutate the site of GPR91 where 3693P might be binding, as shown in red, after mutation, there was no change in the luciferase activity. So this assay was a confirmation that GPR91 is the direct target for microRNA 3693P. So having established these, we were then interested to understand in further depth what's the functional role of this microRNA and if it regulates the succinate GPR91 signaling. So to understand that, we looked at the expression of GPR91 first under oxLDL condition, and you can appreciate that when the cells were loaded with oxLDL, there was significant increase in GPR91 expression. Now, another study has shown that GPR91 activates its downstream inflammasome through NLRP3 and IL-1 beta, as well as our previous reports also suggested that it might be also involved in mitochondrial dysfunction. So we were quite interested to understand these two aspects of the signaling, whether GPR91 is regulating these two pathways in our conditions. So what we found was that while oxLDL loading increases GPR91 expression, as well as the inflammasome and mitochondrial dysfunction markers, when you overexpress 3693P, all these markers were significantly decreased, suggesting that microRNA 3693P overexpression was able to halt the activation of both inflammasome as well as the mitochondrial dysfunction that was being done by succinate GPR91 signaling. And conversely, when we inhibited the microRNA, we found opposite results, that when you inhibit this microRNA, there is huge increase in GPR91 expression, as well as the parallel increase in NLRP3, inflammasome, and mitochondrial dysfunction markers such as ERK signaling and phospho-DRP1. And as a readout of inflammasome activation, cleaved IL-1 beta was also significantly increased when the 3693P was inhibited. So both these experiments clearly indicated that microRNA 3693P was upstream of succinate GPR91 signaling and was regulating the inflammasome as well as the inflammation in the mitochondria, in the macrophages. So to summarize so far, microRNA 3693P regulate immunometabolism via succinate GPR91 signaling. Our next question was whether therapeutic administration of this microRNA can halt the progression of diabetes-associated atherosclerosis, and what are the mechanisms for that? To answer these two questions, we performed mice study where LDLR knockout mice were similarly fed on high-fat sucrose diet for a period of 12 weeks. But when they were at 8 weeks of diet, they were injected with either non-specific control or microRNA 3693P mimic to overexpress this microRNA. So the regimen for the dose there were 2 doses per week for a period of 4 weeks. And we validated the overexpression of this microRNA by fact sorting the macrophages at the end of the study. And we found there was approximately 7-fold increase in the expression of this microRNA in the mice that received 3693P injection as compared to the non-specific control. When we looked at the lesions of these mice, we found that as compared to the control mice, there was a huge decrease in the lesion area in the mice that received 3693P injection. Approximately 27% lesion area was decreased in these mice as compared to the control mice. And these mice also showed less lipid content in the plaques, suggesting that the macrophages were not taking the oxalDL or other lipids in the lesions as compared to the control ones. So because the lipid content was decreased, we then wanted to understand if the circulating lipids or the other glucose and insulin parameters have a role to play in this study. To our interest, we looked at the total cholesterol, triglyceride, non-HDL cholesterol, HDL levels. None of them were changed between the two groups. There were no change in the body weight, no change in the blood glucose. There were no change in the glucose tolerance or the insulin tolerance test, suggesting that whatever changes that were observed in the previous experiment were independent of any changes in the lipid or the glucose parameters. So something at the local microenvironment was happening that was regulating those changes and were being mediated by this microRNA. So to understand the mechanistic aspect of the phenotype, what we did, we took the aortas from the mice that received either the control or 3693P mimic injections, and then we performed aortic digestion and also then fact sorted those macrophages from the cell suspension. And from these macrophages were then performed with RNA sequencing. And what we found was that approximately 7-fold increase in microRNA was observed in 3693P injected mice as compared to the control mice. And from the transcriptomic analysis, to our interest, we were interested in first looking at what happened to the target of this microRNA in vivo. We already validated the expression of succinate receptor 1 or GPR91 in our in vitro studies. Here, we wanted to understand if this was also repressed in the mice that received 369 injection. You can clearly see that mice that receive 3693P injections have significant decrease in succinate receptor 1, which was among the top targets that were being downregulated by this microRNA. We also validated our observations through flow cytometry in both the circulating monocytes as well as the aortic macrophages. And you can really appreciate that the expression of GPR91 was significantly decreased in both the monocytes as well as the lesional macrophages in the mice that received 369 3P injections. We then also wanted to profile the immune cells that were being circulated in these mice. There were no change in the total monocytes in both the type of mice. But very interestingly, there was significant increase in the Ly6C low subset of monocytes. And as a reminder, these are the monocytes that are anti-inflammatory in nature, while Ly6C high monocytes was significantly decreased, and these are the monocytes that are pro-inflammatory in nature. So you can clearly see a shift from pro-inflammatory monocyte to a anti-inflammatory monocyte status of these macrophage monocytes, suggesting that the inflammation was dampened by 369 3P injections in the circulating monocytes. We did not observe any significant changes in other immune cells, such as neutrophils, B cells, CD3 T cells, T4 cells were significantly increased. But we are still exploring why this was, what are the other cells that are associated with the CD4 positive subset of T cells. However, CD8 positive T cells did not change. Because our Ly6C low monocyte in circulation was significantly increased, we were interested in understanding if these monocytes were then recruited to the lesions in the aortas. So some of the mice were injected with a colored beads that you can see in green that will only be specifically being taken by the Ly6C low subset of monocytes. So we can track these monocytes in the lesions. So when we injected these beads in either the control mice or the 369 injected mice, you can really appreciate that the mice that received 369 3P injection had a significant increased number of the recruitment of these beads in the plaques as compared to the control ones, suggesting that what we saw in circulation, increase in Ly6C low monocyte subset, they were actually reaching to the plaque environment, and there were also being increased recruitment of these anti-inflammatory monocytes. And because these monocytes, when recruit to the lesions or plaques, they are then differentiated into mature cells called macrophages. We also wanted to understand what happened to the phenotype of macrophages in the mice that received 369 3P injections. So what we found was that, consistent with the monocyte profile, there was significant decrease in the pro-inflammatory macrophages in 369 3P injected mice as compared to the control ones. And there was a parallel increase in the M2-like macrophages that are anti-inflammatory in nature, also known as pro-resolving macrophages that are known to resolve inflammation. They were significantly increased in 369 3P injected mice as compared to the control mice. There was significant decrease in the markers for the inflammatory macrophages like CD86, while there was significant increase in anti-inflammatory markers like arginase 1 that was increased in 369 3P injected mice, suggesting that there was a shift of phenotype from M1 to M2 state in 369 injected mice, and that these microRNA was able to dampen the inflammation and was able to promote the pro-resolving environment in the atherosclerosis in the diabetic mice. We went back to our transcriptomic data and wanted to understand what are the pathways that were emerging from our data set. And the top pathway that we found that was activated in the macrophages from 369 injected mice was phagosome formation. And just an introduction about phagosome formation is that macrophages are known to eat or engulf the inflammatory or the foreign particles or dead cells, which is where their phagosome phenotype is being activated, and the process is called as phagosome formation. So by doing so, they create a pro-resolving environment in the lesions, and therefore they inhibit the inflammation in that environment. So the top pathway that emerged in our data set was phagosome formation in 369 injected mice. So we then wanted to understand if this was happening at the lesions as well. So to confirm the transcriptomic analysis, we then looked at the ability of these macrophages to clear the dead cells, the apoptotic cells. So that process is known as apherocytosis or phagosome formation. So using immunostaining, what we found was that macrophages from 369-3P injected mice showed a higher number of engulfment of dead cells as compared to the control mice, suggesting they were having higher apherocytotic ability as compared to the control mice, suggesting that they were able to clear the dead cells more efficiently, and there was therefore reduced plaque burden in these mice as compared to the control ones. And consistent with this assay, we also looked at the necrosis in these lesions, and you can see that as compared to the control, where you can see huge spaces of the necrotic space, these are the spaces where dead cells just sit there and create more inflammatory environment. The mice that received 3693P injection, the necrotic area was significantly reduced, suggesting these plaques were shrunken in size. Their dead cells were more efficiently being removed, and there were more stable plaques in 3693P injected mice. Lastly, we were also interested to validate our in vitro findings, where we found that 3693P was regulating GPR91 expression and therefore regulating inflammasome through NLRP3 signaling. So in vivo also, we wanted to validate these findings. So through immunostaining, what we found was that firstly, there was significant decrease in the macrophage content in the mice that received 3693P injection as compared to the control one. CD68 is a marker for macrophages, and you can really appreciate that as compared to the control mice, there was significant reduction in the macrophage burden. And in these macrophages, when we further probe the NLRP3 expression, you can see there is a huge decrease in the inflammasome expression in the mice that receive 369 injection as compared to the control one, suggesting that 3693P was able to halt the inflammation in the diabetic associated atherosclerosis by dampening the inflammasome signaling through GPR91 targeting. So the take-home message for this study is that we found for the first time that microRNA 3693P is downregulated in mice, as well as we found that in the human. We also found that microRNA 3693P regulates the metabolic status of macrophages, converting them from pro-inflammatory status to the anti-inflammatory status. And it was doing so via succinate GPR91, NLRP3, inflammasome signaling. And when we therapeutically restored 3693P injection in diabetic mice, it was able to halt the progression of atherosclerosis, and all those effects were independent of any circulating lipids or insulin resistance. This work was last year well received and was published in Cardiovascular Research. And based on this work, I was also awarded American Heart Association Career Development Award last year, where my current interest is broadening into studying the role of this microRNA in regulating immunometabolism, this time in regression of atherosclerosis in diabetes, which is so important that we cannot just rely on treating the patients just that they will develop atherosclerosis. Most of the patients have already developed atherosclerosis, which is highly burdened in diabetic subjects. So the next area of my research is focused on how we can regress those plaques that have already been established in this population. With that, I would like to extend my gratitude towards my mentors from my PhD in New Zealand, as well as my team there. all my postdoctoral mentors from New York as well as from Harvard, and my collaborators and the patients who participated in the studies and all the funding agencies to fund this research. With that, thank you so much for listening, and I'm happy to take any questions.
Thank you so much, Dr. Shruti Rawal, for delivering such a compelling and insightful session on microRNA regulation of diabetes associated atherosclerosis. Your presentation beautifully highlighted the complex interplay between microRNAs, inflammation, and vascular dysfunction, and offered a deeper understanding of their potential as therapeutic targets. We truly appreciate the clarity with which you conveyed such intricate scientific concepts and the passion you bring to your research. Your work is not only advancing the frontiers of cardiovascular science, but also inspiring the future direction in translational medicine. Thank you once again for your time and exceptional contribution to our 88th SPSR International webinar. Ma'am, some questions are there from the participant side, and I will be asking you one by one.
Sure.
So the first one is, which specific microRNAs have been most strongly implicated in regulating atherosclerosis in diabetic conditions?
There are only handful of microRNAs that have studied in this field. So this field is really evolving. But one particular microRNA that have been studies is microRNA 33. And then the second one is the current microRNA that I just presented. Yeah.
Right, ma'am. Thank you so much. The second one is, how do microRNAs influence endothelial dysfunction in the context of type 1 and type 2 diabetes?
That's a very good question. Um, so microRNAs, as I just presented, they function by regulating the targets. So if these targets are involved in several pathways, for example, in endothelial function, nitric oxide synthase plays an important role. So there are several microRNAs, including microRNA 126, which I have studied in my PhD, that is known to regulate these targets. And by doing so, they can then regulate endothelial dysfunction in type 1 as well as in type 2. Although in type 1, there is autoimmunity, and the type 2 has the metabolic dysfunction, but the overarching mechanisms remain the same. That is inflammation, where nitric oxide plays role and microRNAs regulate these pathways. Yeah.
Right, ma'am. Thank you so much, ma'am. The next one is, ma'am, could you please explain how micro RNA and mRNA interactions alter the immune metabolic pathways in atherosclerosis?
Yeah. So microRNAs, it binds to the 3' UTR sequence in mRNA and therefore degrades its expression. So these mRNAs are, if they are involved in several metabolic status of immune cells, and if microRNAs are targeting those mRNAs, it can then influence the immune behavior. For example, there are microRNAs that are pro-inflammatory in nature, such as microRNA 155. It regulates SOX1, which is known to regulate cytokine production and therefore enhance the inflammatory response. So there are these microRNAs are in the category that can enhance the inflammation response. But there are other microRNAs like microRNA 146 that is known to suppress inflammation. So by regulating different or distinct targets, microRNA can fine-tune the immunometabolic status of cells. Yeah.
Right, ma'am. Thank you so much, ma'am. The next one is, what interdisciplinary approaches or collaborations have enhanced your research outcomes?
Oh, there are many. But to name few, on daily basis, I collaborate with computational biologists. I heavily rely on them for looking at the targets, looking at the transcriptomic analysis pathways. So we have a team of computational biologists that we heavily collaborate. In addition to that, in US, there is a culture to have collaborations with clinicians. There are several sites where you can actually collaborate and request for clinical samples, and they're known as consortiums. And one such consortium I recently joined, where I'm seeking samples from type 1 diabetic patients to further enhance my research. Yeah.
Right, ma'am. Thank you so much. The last one, ma'am. What are the key questions your lab is currently working on in the field of diabetic cardiovascular complications?
Right. So my lab, the research theme of my lab is two arms. One is atherosclerosis that I have presented. Another one is looking at the ischemia, hind limb ischemia or myocardial ischemia. So these are the two basic directions my lab is currently focused on. Yeah, and the center for both these processes are non-coding RNAs, but either it is microRNA or it's long non-coding RNAs.
Right,
Yeah.
ma'am. Uh, Thank you so much, Dr. Shruti Rawal, for answering all the questions so wonderfully and with such depth and clarity. Your thoughtful responses have truly enriched our understanding of the complex regulatory roles of microRNAs in diabetes-associated atherosclerosis. We sincerely appreciate your time, patience, and the way you engage with each question so thoroughly. It has been a privilege to learn from your expertise during today's session. Thank you once again for your outstanding contribution to our 88th SPSR International webinar. So here we come to the conclusion of this session, ma'am. As we conclude the 88th SPSR International webinar, we extend our sincere gratitude to Dr. Shruti Rawal for her outstanding presentation on microRNA regulation of diabetes-associated atherosclerosis. Today's session offered us a deep and detailed understanding of the molecular and immune metabolic pathways influenced by microRNAs in the progression of vascular complications associated with diabetes. Dr. Rawal's work beautifully demonstrated how cutting edge techniques ranging from RNA transcriptomics to mouse models and metabolic profiling can be harnessed to unravel new therapeutic avenues. Her
Thank
insights
you so much for.
not only highlighted the critical roles of microRNAs in endothelial dysfunction and inflammation, but also pointed to promising translational strategies that could revolutionize cardiovascular care for diabetic patients. On behalf of the Society of Pharmaceutical Sciences and Research, we thank Dr. Shruti Rawal for sharing her expertise, and we appreciate the engaging participation from our attendees. We are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your significant input. We would also like to express our appreciation to our convener, Mrs. Monika Sabharwal, for her pivotal role in organizing this event. We extend our gratitude to all the SPSR team members for their collaborative efforts, which have broadened our understanding and fostered cooperative learning. Your collective contributions have made this webinar a success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. We look forward to your participation in future SPSR webinars as we continue to explore groundbreaking topics and foster knowledge sharing in pharmaceutical sciences. Thank you once again, and we wish you all the success in your endeavors. Have a wonderful day ahead. Thank you so much, ma'am. Thank you from Team
Thank
SPSR.
you. Thank you so much for having me. Thank you. Bye. Take care.
Thank you, ma'am.